This is the 9th session of the conference, third day, etc. And we start off with Professor Sitabhra Sinha from IMSC, Flags, Landscapes and Signaling.
Okay, so thanks for the introduction, and let me first of all express my gratitude to the organizers, especially Nilima. Ram showed this photograph of the first PNLD 2004. So that actually is from my institute, the Institute of Mathematical Sciences. So we actually co-organized it with IIT M, and Shudeshna, who at that time was my colleague at IMSC, was helping me to organize, like show me the ropes, how to actually, run big conferences. So she was like the lead and I was like the co-lead, doing these things in IMSC. So it's a great pleasure to see PNLD back in Chennai. So I'll be talking about some of the very recent work that this freshly graduated PhD student from my group, Chandrasekhar, and senior scientist in my group, Dr. Shakti Menon, has been doing. In order to explain what this means, let me just go straight to what's the problem that we are trying to solve. So essentially, this is the question. All right. So how do you start from single cell to, let's say, this, or maybe all these creatures who are sitting in these seats, right? Or for that matter, a mouse, a dog, whatever. All right. So the interesting part is that every cell of this animal's body has exactly the same genome. Okay. So this is like to counter, like the usual, gut reaction. If you ask someone, okay, how do you go from here to here? You say, oh, it's in the genes, right? Well, yes, but no, because every cell has exactly the same gene, and yet, it's not exactly a homogeneous mass, right? Every cell is distinct. Moreover, every cell knows its position. You don't have eyes sprouting out from the legs, or you don't have a tail kind of coming out of its nose, right? So how does that happen? If you think deeper about it, you realize the problem is even more complex, because what are actually genes doing? Genes are actually coding for proteins. So you have, let's say 30,000 genes, and each of them, let's say, not all of them code for proteins, but let's say each of them are coding for proteins. Well, you have so many different types of proteins, but then somehow it has to all come together to form this very characteristic shape, which you immediately recognize as the tiger or a human. Right? So what tells the proteins to self-organize themselves into structures, and then forming higher structures, and those higher structures can then self-organize so that eventually this thing that you see in front of you can actually move its mouth and make noises. It's an incredible problem, and of course one can only hope to try to make a small dent at some aspect of this problem. Just to illustrate like where exactly we are trying to strike at the problem, let me show you this fantastic movie, which is essentially done by this group at University of New South Wales. So this video shows the development of the zebrafish from the single cell state. So basically, when we all start life as a single cell, the egg, and when it's fertilized, eventually something happens and that single cell starts dividing. So it becomes 2 cells, then the 2 cells become 4 cells, 4 cells become 8 cells, very much like period doubling, right? Except that this is in space. And for a time, you will see that it will look like just one giant homogeneous mess of cells. And then something happens, some cells start differentiating, and they would express certain proteins and not express other proteins. Other cells would, express a completely different set of proteins. And then you will start seeing, elongations happening, a distinct head forms, a distinct tail forms, and so on. Okay? So let me just show you the movie. All right. So here is time, and you can see that it's way accelerated. So it's basically showing every 40 seconds of developmental time from 30 minutes post-fertilization to 12-hour post-fertilization, and then after that it's showing 80 seconds all the way up to 24 hours post-fertilization. So you can see it still looks very much like a sphere, except that now something like a snake-like shape is forming. You can see something which looks like a head and a tail. Okay, so this is the head part. So then the tail separates from this yolk-like object. You can see the beginning of the backbone. Okay, so this vertebral structure. This is now starting to look very much like a fish larva, right? And then you can see, the part which will eventually become the fin or the tail. This yolk-like, sac-like thing gradually gets absorbed into the body. And then by the time the movie ends, which is 85 hours post-fertilization, you will see something which looks very much like fish baby. All right. So the fundamental question we as physicists are asking, so we are not biologists. We don't look at the molecular biology details of what's going on. What we're asking are, so you start with something which is perfectly symmetric, right? So the Greeks had this thing about a perfect shape being a sphere because it had all the symmetries, right? So the egg is almost like this perfectly symmetric shape. And yet, the thing that you see at the end of this movie is something which is anything but symmetric, right? So all the symmetries that were there originally are lost. And so we are asking the question, what are the mechanisms which lead to the symmetries of this initial state being broken? So that's where we as physicists come in, because if it's one thing that physicists are kind of really obsessed with, it's how do symmetries break? So bifurcations that we all talk about is one of the mechanisms by which we know that various symmetries can be broken. And, there have been a large number of mechanisms which have been proposed, but they can broadly be classified into two main classes. Right? So in order to explain how an initial homogeneous object can end up with various kinds of broken symmetries. Patterns are nothing but broken symmetries. Usually people either appeal to one of these two. So this is a self-organized mechanism where chemicals react with each other and also diffuse in order to form patterns which look like spots or stripes and so on. And the very famous Turing mechanism is actually one of the examples of such self-organized pattern formation. But if you look at a book on developmental biology, you'll find that this is almost very rarely mentioned, if at all. A typical book on developmental biology would more or less focus on a completely separate class of mechanisms, which one can call boundary organized, and I'll explain what boundary organized means within the next slides. So here, there is a chemical cue, okay, which is, because of many reasons, is called a morphogen. So that morphogen basically diffuses from wherever it's being produced, and as it diffuses, it creates a exponential gradient, concentration gradient. And depending upon the concentration at various points, cells at different locations switch on different programs. Okay? And that's how cells, for example, closest to the morphogen source expresses a particular fate, which we can call the blue fate. cells over here express what we might call a white fate, and cells over here express what we might call a red fate. Okay. So this is different from this because over here there is no such external signal which is being used. Okay. Here the reactions are all occurring between the players themselves, whereas here the players are actually listening to some commands which essentially is coming from outside. Okay. So a little bit about each of these mechanisms. Okay. So boundary organized pattern formation, key idea is that of something which is referred to as positional information. Okay. So what does this mean? It has essentially two elements. One is the cells receive some external cue, so for example the morphogen concentration, which tells them where are they in the body. And you know, if you want to know where you are, you need some reference point, right? So you need to know, okay, this is the origin, and with respect to the origin, this is where I am. Otherwise, if I just give you a set of coordinates, that's not going to help you much. You also need the origin. Okay? And so typically here, the reference point is the boundary of the tissue or the organism or whatever. And that's why it's called boundary organized mechanisms. So with respect to the boundary, which acts as the reference, you basically get information about where you are. How far are you from the boundary? Okay? So you now know location, but that's not enough. Now knowing this information about where you are, you decide to initiate a certain set of actions. So depending upon your position, you will initiate program A or program B or program C. Now, the interesting part is these two need not be related. Once you know your position, at different times, you can actually switch on different programs to do completely different things. Now, to illustrate this, let me show you this is the Arirang Mass Games, I hope I'm pronouncing it right, from North Korea. And let me ask you to focus not on what's happening on the field, but what's actually happening among the spectators. So you can see, you know, all these flags. So how are these flags being created? Any ideas? So people are just holding up placards, right, of different colors. Yeah. Now, have that pattern. It's because people who are sitting here have been given placard of a particular color. People sitting over here have been given placards of red color. People sitting over here have been given placards of white color, and so on. So the positional information is when they go into the stadium, they know which seat they are going to, and by looking at the seat number, they are being given boards of various colors. Okay? Now, you might say that, okay, now the program is kind of tied to where they are sitting. But this is the interesting part. They can have multiple boards, right? And using multiple boards, they can actually create very different images. So you will see that, you know, first they have this flag, and then after a while it will change and they will create a different colors because they're going to switch on a different program altogether. Actually, this is pretty spectacular. I mean, what's happening in the field gets paled by actually what's happening on the stands. Well, so is your body. Well, you know, in Asiate they tried to do that, in Asiate 82. So yeah, so now you can see a close-up, right? And you can see they just changed the, and you get a completely different pattern, okay? All right, so now it looks completely different. Okay? So this is pretty much what's happening. Sorry about that noise. So actually they are doing calligraphic patterns now. So if you look at it from far away, it's basically some calligraphic messages. Okay, so you get the gist of what I'm going to talk about. So then, we understand that by having this information about where you are, you could actually have the ability to switch on different programs and thereby decide to express different proteins. Okay, so in principle now we have this idea, okay, if I am like close to this point, maybe I would switch on some proteins which will make me neuron. If I'm here, maybe I'll switch on some proteins which will make me muscles and so on. Okay. Now, the first model which kind of showed how this can happen was proposed by Lewis Wolpert, who's actually unfortunately passed away a couple of years during the COVID epidemic. So he kind of gave a very, very simple model which essentially said that, look, diffusing from a source, then the concentration gradient kind of would be exponential or linear or whatever. And then the cells over there, so these are the various cells, which initially were not differentiated at all, depending upon the morphogen concentration locally, will switch on different genetic programs and thereby go to certain fates. So for example, this will choose the fate A, let's say they are neurons. This will choose fate B, let's say they are cartilage or whatever. This will choose fate C, and this will eventually become muscle or whatever. Okay. Now, this we realized can be tied beautifully with something that is actually very, almost contemporaneous. So this is Conrad Hal Waddington, who was a biologist working on developmental pattern formation, but he is also very famous for having promoted the idea of theoretical biology. So in the 1960s, he actually sponsored a series of conferences called Towards the Theoretical Biology. And in his book, The Strategy of the Genes, he actually gave this very influential figure. So now you see this figure reappearing in lots of papers in Nature, Science, and so on when people are talking about epigenetics and so on. So the idea that he had was that when a cell starts life, it has an unlimited potential. It's pretty much like each of us, right? So when we are kids in primary school, we think we'll become astronauts or generals or president or whatever, right? The world's our oyster. Okay? But then as time proceeds, as Puneet said, life happens, right? And you start going through a series of so at each branch point you choose left or right, and that immediately constrains your choices. Okay, so here for example, as it goes down, you could eventually end up in the blue fate or the white fate or the red fate. Okay, so like many of us, let's say I wanted to be a rock and roll singer when I was in class 6, and then in class 10 I said, okay. Maybe I'll take science, and then in class 12, okay, maybe I will take physics, and then after bachelor's I said, let's try taking pure physics, and then after a while you take a series of choices after which you are no good for anything except becoming a professor at the Institute of Mathematical Sciences. Very similar thing happens in the case of a cell. So once it differentiates and forms this final thing, it's not really possible for it to go back and start doing something else. Of course, now we know that you can induce pluripotency. So it's like getting a second chance in life, right? So at the age of 60, you decide, okay, maybe I can still become a classical guitarist, okay, and start a second innings. Right? But for cells, you need to do a lot of perturbation for this to happen. And usually, in normal circumstances, this doesn't happen. And Waddington basically said that, look, this landscape, if you think of it like a tent, very corrugated tent, then something's holding it up, right? Something is giving shape to these corrugations. So this, huge assortment of tent pegs and ropes, which is kind of causing this architecture, is basically the set of genetic interactions. So there's, as they say, like when you see the swan gliding by, there's a lot of furious paddling going on underneath. So the furious paddling over here is being done by this set of genetic interactions. So far, so good. So this is at the level of a single cell. And so we started thinking, okay, what if we combine Wolpert's idea with Warrington's idea? So essentially what is happening is that if you think of this picture on a tissue, on a spatially extended system. So the cells over here, something is happening such that the blue fate is kind of biased very strongly. And so instead of having, all of these things kind of being almost similar depth, let's say the blue well is becoming so deep that it's very likely that the cell is going to end up in the blue fate. On the other hand, at this end, because of the higher concentration of the morphogen, something is happening such that the red well is becoming way deeper, and so it's much more likely that the cell is going to end up here, and so on. Okay? So the question was, how do you actually tie up the set of these genetic instructions which is happening at the scale of a single cell, genetic regulations which is causing these conditions, to bias it such that certain attractors become much more likely than other attractors depending upon where you are located on a spatially extended system. Okay? So that, in a nutshell, is this problem of pattern formation in embryo that we are trying to solve. Okay? Now, the main problem that we see with Wolpert's idea is that if you don't assume any noise, it works beautifully. Unfortunately, biology is just full of noise. And so what we realized is that, so if you look at something like this, so this is basically our model of a vertebrate neural tube. So this is, sure about that? Okay. So if you look at the vertebrate neural tube, so you can see that you have the red fate over here, white fate over here, blue fate over here. But under different conditions, you can see that you can end up with like a all red flag, very familiar from the state where I come from, at least a couple of decades back. Or you could have something which looks like mostly white with a bit of red thrown in, and so on. So how is it that despite so much noise, you don't end up like this, but mostly end up with this? I mean, it will be rather unfortunate for the organism which ends up with something like this, right? And so for this, we took from the other set of pattern formation mechanisms which has been proposed. As I said, Turing is responsible for initiating the idea that you can take activators and inhibitors, have them react with each other, and if the inhibitor diffuses much faster than the activator, then you have spontaneous formation of spots or stripes. So this can be much generalized by saying that what's the mechanism is basically local activation, lateral inhibition. And there has been a lot of debate until the 1990s about whether Turing patterns actually form in biology. So it was only in 95 that Sai actually showed that Turing mechanism is the only thing that can explain, for example, these fish stripe patterns in, for example, Pomocanthus. And now, of course, a lot of other examples have been shown to work. And in fact, the general concept of lateral inhibition, we know, actually works in lots of different systems, including the formation of orientation columns in the visual cortex. And in fact, in the context of development, the idea that lateral inhibition is at work predates Turing. So Wigglesworth in 1940 had actually used the idea of lateral inhibition to explain how these bristles form in the underside of insects. So what we are working on is to understand how you can have lateral inhibition implemented in the real context of development. So most models of lateral inhibition uses diffusion. Okay? Unfortunately, it doesn't work if you have a multicellular organism simply because the cell wall acts as a big hindrance. You have to work with something like either gap junctions, which are specialized proteins which allow things to go through them, or you could have something like a chemical reaction between the surface receptors and surface bound ligands. Okay? So this is essentially what we are talking about or what we are working on. Particularly, we are looking at the notch pathway. Now, there's a usual, like a statement made by many biologists that there are two kinds of biologists: those who are working on notch pathway and those who don't know they are working on notch pathway. It's that important. Okay? So essentially, notch pathway can actually replace diffusion, and you can still have basically the similar kinds of pattern formation. So what we're asking is that could this kinds of intercellular communication through notch pathway affect the French flag to make it, for example, the flag of San Marino, or that of Monaco, or that of Liechtenstein? Okay? So we have been working on models where we have this blue, white, and red genes which are regulating each other. So this is one cell, this is another cell, and they are talking to each other through this notch receptor and ligand interacting. And as a result of these interactions, we are showing how you have high concentration of blue appearing at one end, red appearing at the other, and white appearing in the middle. Now, Nilima always complains that I never show my equations. I always tend to be only figures. So because Nilima is in the audience, I was like, I have to have some equation. So so these are the equations which regulate how the concentrations of blue, white, and red actually grow with time. But you will notice that they also involve the ligand concentration and the notch receptors, and that's where the coupling between the cells are actually coming in. All right? And so what we find is that certain kinds of interactions are much more likely to promote the flag, the blue, white, red kind of thing. Certain other kinds of interactions to extremely bad flags like only white or white and red and so on. Look at all kinds of interactions which happen, and you can find that certain kinds of interactions are pretty bad. And then you say, okay, so what you have shown is that intercellular interactions do affect the flag, but so what? So it turns out that you can show that this contact-mediated cellular interactions can actually increase the precision of the patterning subject to noise, very surprisingly. Okay? So for example, if in the idealized case, you would expect that cells over here should all express the same fate and all express here the orange fate. But in reality, because of noise, you could have crazy jagged boundaries, right? How is it that you get this in this? You have to have some kind of a noise reduction mechanism. Again, we work on something a little simpler because we just have to have two types of genes expressing fate A and fate B. And what we show is that if you have no coupling, then you have this very broad distributions of fate patterning. So this A is peaked over this broad range, B is peaked over this broad range, and you have the cells, for example, in the middle. By chance, they can sometimes show blue fate, sometimes show orange fate, and so on, and you can have all kinds of crazy jagged patterns. The moment you have coupling, you can see that this turns into an almost step function-like behavior, and the cells at the boundary know exactly whether they have to be blue or orange, and you have a very, very sharp boundary. Okay? Now, I'd just like to end by showing that there's a very interesting analogy of all this with a model that we were very familiar with in physics, that of collective ordering. Okay? So if you look at the Ising model, you have typically the coupling between through the exchange interaction, and this is actually analogous to the cell-cell interactions through notch. On the other hand, you have this external field which is acting as the environment, and this field is actually its role in developmental biology is taken by the morphogenetic field, right? And just as you have by changing temperature, by changing field, you can get regimes of order and disorder. We can show that if you have a spatially varying external field. So this is basically analogous to the morphogenetic field, which is decaying exponentially. So if you have no noise and if you have no interactions, then of course this field is just going to create an all black pattern here, all white pattern here. But the moment you switch on noise, you can see that it's completely a mess. And now, if I switch on interactions in the presence of noise, you can see some order is coming back. And finally, when I have very strong interactions, I get something which is almost close to this, the perfect pattern which was not there for the noise. And so, to end, from the developmental landscape, we essentially can think of, this is as analogous to something like an energy landscape, where the pattern that is being formed is basically the flag, and the morphogen gradient is actually acting as the signal, and this is analogous to the field that we are familiar in statistical physics. Okay, so let me just end by thanking the people who actually did this work. So as I mentioned, this is the doctoral work of my grad student Chandrasekhar, who is currently a postdoc at Harvard and Howard Hughes Medical School, and Dr. Shakti Menon, who is a senior group leader in my institute. And of course, I'd like to acknowledge very generous funding from the Department of Atomic Energy. Thank you.
Thank you, Sitabhra.